Hi, everyone. So I'm George DiCenzo. I'll be the host for this last session of the day, which is on natural products and enzymes and is sponsored by the NURCASA project. So we'll get started with Rachel Keown, who's from University of Delaware, and will be telling us about her talk, Novel Viral DNA Polymerases from Metagenomes Exhibit Diverse Biochemical Phenotypes.
Thanks for the introduction, and thank you all for hanging in this afternoon to listen to my spiel today. I'm in my third year of PhD study at the University of Delaware, and I'm here today to talk to you about some work that we recently published in a manuscript in Frontiers in Microbiology. I'm obviously very excited about this work, so I hope that by the end of this, you will be too. So first, just some general background about the lab. We go by VAIL, or the Viral Ecology and Informatics Lab, and we are quite a large group at the University of Delaware. And all the friendly faces you see up here are all of my lovely coworkers. Since we are such a large group, our work is pretty diverse. I work on the wet lab side of things, which I will tell you more about in just a moment. But we have students studying things ranging from soybean bradyrhizobium to the oyster microbiome and even some viral dark matter work. We also have some students that are predominantly in the dry lab bioinformatics world, which is a massive help to someone like me who has had all of their formal training on the bench. But the real benefit of this working group is the fact that we have 3 different PIs that have different expertise across a few different fields. Sean Polson has a background in microbiology, and he is also the director of our computational biology core facility at University of Delaware. Eric Womack has expertise in the world of microbial ecology, and Jeff Furman has expertise in soil science and microbiology. So this diverse grouping of PIs really lends itself well to all the different projects that we have going in the lab, which I think is pretty awesome. So even though we study a few different kinds of systems that are pretty diverse, what really unites us are the questions that we ask of those systems. We're mostly interested in viruses that infect bacteria or bacteriophages, and I'll be using phages for short. And the questions that we ask of them include who's there in the environment, how abundant are they, what might they be doing to influence the ecology of their hosts and the environment. And viral infection characteristics or phenomes affect their impacts on hosts and the environment, but that phenomic data is really difficult to generate. However, thankfully, the phenome is encoded by the genome, and thanks to sequencing, we can readily get that genomic data a lot more easily. So our primary area of research is establishing these genome to phenome connections in many of the different systems that we look at so that we can predict how the genotype of an environmental virus can tell us about the phenotypes and the impacts of viral infection. You might be asking why we even care about phage at all. They are the most abundant and diverse biological entities on the planet, with an estimated 10 to the power of 30 viruses just in the world oceans. This number is sometimes a little bit hard to wrap your head around, so my favorite way to think about it is that there are more viruses in just one handful of seawater than there are people on the planet. It's kind of an astronomical amount, right? More viruses than even the number of stars in the observable universe. So even more than their abundance, they're also responsible for a lot of different processes. For one, horizontal gene transfer with their host bacterium can potentially enhance the pathogenicity of the host. Viruses will also drive biogeochemical cycling, especially in the open oceans, and they'll also shape their microbial host populations by way of infection and lysis, recycling some organic matter back into the environment. So what this all means really is that viruses have a huge impact on the global carbon and nutrient cycles. So a real-world example of phages impact is exemplified by the bacterium Prochlorococcus. And this bacterium is highly abundant in the world oceans, as shown by the heat map on this slide here. These bacteria are responsible for up to 20% of the oxygen production on Earth. And by extension, the cyanophages that will infect these bacteria also have an impact on that process. Specifically, many of these phages carry auxiliary metabolic genes, or AMGs, that will actually alter host metabolism to help produce more phages during infection, ultimately impacting the amount of oxygen produced by the bacteria. Another more general example of phage importance is that of the viral shunt. This refers to the process by which phages feed into that dissolved organic matter pool within an aquatic ecosystem. And the main arrows here of note are in red. And it shows you how phages can contribute to this pool in 2 ways: by infection and lysis of both phytoplankton and bacterioplankton. This means that viral infection can be responsible for up to 50% of that dissolved organic matter pool, highlighting the importance in the ecosystem. This figure also shows the different life cycles that phages can exhibit, and each has very different biological characteristics. So the lytic or the virulent lifestyle, shown up at the top here, is important for production of high numbers of progeny, as well as those contributions to the bulk organic matter I just mentioned. And these viruses will replicate very quickly. Lysogeny, or the temperate life cycle, shown down at the bottom here, is less about the production of progeny, but more about the survival in certain environments, like those that might not be suitable to reproduction of either the bacterial host or the phage itself. And these viruses will integrate into the genome as a prophage here and remain dormant until lysis is triggered, typically by some sort of environmental factor. There's some nuance here, though, that every phage is different, and instead of these life cycles being two distinct ones, you actually get varying levels of infectivity, latent period, and production of progeny. That kind of brings us to the problem of phenotypic data collection. Traditionally, scanning electron microscopy was used to classify some viruses by their morphology, but it's time-consuming and expensive and doesn't always directly equate to the life cycle characteristics of the phage. So that brings me to some of the difficulties that we face. Environmental phage are really, really tough to study in isolation. This is mostly due to the fact that they can't be cultured in the lab, and you need a specific host for each individual phage. And things like the classical one-step growth curve experiment for phage infection phenotypes are really labor-intensive and really time-consuming. However, genotypic data are a lot more readily available than phenotypic data. Unfortunately, though, viruses do lack a universal marker gene. But on top of that, viral sequences are commonly underrepresented in published sequence databases. So despite all the importance that I just mentioned about all of these abundant phages, we really don't know a lot about phage biology. So you'll remember this figure that I showed earlier. We kind of have to build our own framework of studying environmental phages. So we use this genome to phenome approach a lot. And we do our best for genotypic characteristics, even though we don't have a universal marker gene like that of 16S for bacteria. We do our best to use genes that are pretty common in phages. And one of those genes is DNA polymerase A. The slide shows the structure of DNA polymerase. And this is carried by about a quarter of double-stranded DNA viruses. While that is not the majority, it is a grand chunk of them. The structure here is pretty distinct in viruses from cellular organisms, though it is also carried by cells. In cells, you'll actually have an extra exonuclease domain, which moves in the 5' to 3' direction, responsible for the removal of Okazaki fragments. But in viruses, we only have the 3' to 5' exonuclease domain that is commonly used for proofreading. This enzyme is really diverse in viruses, which I'll show you on a tree in just a few minutes, but prior in vitro mutagenesis studies have shown on this enzyme that the 762 residue that I have labeled here is actually responsible for the incorporation of DNTPs. You can see that it sits right down here where the DNA kind of hangs out. So that's pretty logical. And changes to that 762 residue through those mutagenesis studies showed that it will alter the speed and the fidelity of the enzyme. And these were done in just a few model polymerases like that of T7 and E. coli. But previous metagenomic studies from our lab have shown that viruses can actually have 3 different residues at this 762, which I'll talk more about in just a moment. Before I get to that, though, I just want to remind you of the functions of DNA polymerase, the first being primer extension that I have shown here. The enzyme will bind to a DNA-RNA hybrid, RNA being the primer, and it will extend that primer by incorporating DNTPs and replicate the DNA, creating a double strand. Another somewhat lesser known function of DNA polymerase is called strand displacement. I find that it's easiest to think about this in terms of a circular double-stranded DNA as shown down here. Basically, you'll get primer extension going first. It'll continue replicating all the way around until it gets to the very beginning where the primer was. And it will actually, the enzyme will actually displace that previously replicated strand and continue replicating. So at the end, you basically have a really, really high molecular weight piece of DNA. And I can show you that visualized on a gel in just a moment. But before I get to that, I want to get back to those 762 residue identities I just mentioned. As I said, metagenomic analyses revealed that phage can carry one of 3 residues at this site. The cellular wild type is phenylalanine. This is carried by most bacteria, but also carried by many phages as well. And for the remainder of this presentation, phenylalanine will be coded in red for all of the trees, and you can see it's up here at the top in this tree. Tyrosine, which is shown in blue in the middle here, is carried by many phages, including that of E. coli phage T7. And then leucine in purple is carried by many known lysogenic phages. Prior work in our lab hypothesized this residue can be indicative of phage lifestyle and replication strategy. And this phylogeny shows you metagenomic POLA sequences along with some known phage representatives. But what's really striking about this tree is that this coloring I have here is just by one single amino acid residue in about a 900 to 950 amino acid protein. So to summarize those in vitro mutagenesis studies I mentioned just before, the wild type that they used was phenylalanine because they used the E. coli polymerase. When they mutated that to tyrosine, they saw a 5x increase in the speed. But when they mutated it to leucine, they saw a very large drop, about 1,000 times less speed than that of tyrosine. But the researchers did note a increase in fidelity, which means that the leucine residue is less likely to incorporate mistakes into the DNA. I mentioned that this tree also has some known phage representatives in it. And as I mentioned, they clade pretty consistently with phages that have different life cycle characteristics. So the wild type phenylalanine will cluster with some known lytic phages in here. Tyrosine, on the other hand, clusters with exclusively lytic phages, and then leucine has clustering with lysogenic phages. So in the light of the mutagenesis and the clustering information, it's pretty logical that a quickly replicating phage would need a fast polymerase in order to replicate quickly. And maybe a lysogenic phage that has a more accurate leucine residue is better situated given that they're replicating more slowly, and they may not want or prefer as many mutations incorporated like that would be with a tyrosine polymerase, since they'll have lower population numbers than a phage that's replicating more quickly. So backing up a little bit from one single residue and one single enzyme to talk about viral genomes, viruses carry many more things than just POLA. And a few studies in the lab have shown us that when we use more than just one gene, we can get a lot more data out of our metagenomes. So due to technological limitations like sequencing capacity, we usually look at things that are in pretty close proximity to one another on the genome. The cool thing about viruses is that their genomes are very compact because they're under strict selective pressure, especially for phages like T7 that's shown down here. So you'll see that viral genomes are typically arranged sort of like this. So you'll have your early genes, genes of DNA replication, and then structure and assembly genes. So the cool thing about T7 here is that these DNA replication genes, gene 4 here is a dual primase helicase enzyme, and then gene 5 is the POLA that I was just telling you about. So we know that genome replication is essential to phage survival and fitness in the environment. So genes that are used in replication are a pretty good tool to study lifestyle and hopefully phenotype. Yeah, so after we saw that this association was pretty common in not only known phages, but we see it a lot in our metagenomes, we decided that more DNA replication genes would probably be a good thing to kind of hunt down. And my pet project has kind of been helicases. I worked with helicases a lot in my master's project, so I'm trying to kind of get the relationship between helicase and polymerase to come together. So some additional evidence that this module approach is promising came from a study out of the lab in 2018. So this tree is pretty similar to what I just showed you, except you'll see now that we have 2 splits. We have 5 distinct groups. The phenylalanine group is split into 2 and leucine is split into 2. So you'll see that these groups were actually defined by their replication module content. So you can see that phenylalanine polymerases will associate sometimes with what's called RNR, or ribonucleotide reductase, which is the enzyme responsible for creating DNTPs, or a pool of DNTPs in the cell for replication. As well as you see a few different kinds of helicases that are associated with these phenylalanine polase. You've got DNAB, SNF2, UVRD, and some others. So the interesting thing about this paper is that a student actually dug through all of these metagenome contigs by hand to annotate what the genes were on them. Right, lost my place. Yeah, so the leucine groupings also have a few common associations with helicases, but most often with this SNF2, you'll see here in blue. I'll talk more about that in just a little bit, but just briefly, SNF2 is not an actual bona fide helicase as it cannot separate DNA strands. But also of note, in the leucine groups, we did not see any association with RNR. And then lastly, our tyrosine group. This is most often associated with this GP4 helicase and also commonly has RNR. So we hypothesize that these are highly lytic phages by their replication model content. So this study found a few things. The combination of two genes gave a better phylogeny than just one alone. And additionally, ring-shaped helicases, which are faster and more proliferative, were shown to occur more often with the tyrosine and the phenylalanine groups. And then non-ringed helicases, which are the slower type of helicases, were more often associated with leucine, which would describe a putative lysogenic phage. So just a quick background on helicases. As I said, this is kind of my pet project in my master's. Most of the time we think of DNA as unwinding most of the time we think of helicases as unwinding the DNA double helix as shown in this figure here. But that's not exactly the whole story. Take that down so you're not distracted by the movement. There are actually 6 distinct superfamilies of helicases divided based on their oligomeric structure and specific cellular function. I think as of 2011, the human genome was estimated to encode about 95 different helicases, which really just underscores the fact that they're a really complex grouping of enzymes. So lots of things have the ability to bind to DNA, but in order to be considered a bona fide helicase, you have to have these 3 common abilities. First, obviously, is to bind DNA, which is done by those Walker A and B motifs. Then you have to be able to bind an NTP and use that NTP to drive the unwinding of secondary structures in nucleic acids. As I mentioned before, the SNF2 enzymes that we see commonly annotated in our metagenomes do not have all 3 of these characteristics as they cannot separate the nucleic acid strands. So we do not consider them true helicases. But to get into a little bit more of functional metagenomics, I think you might need a little bit of background on how we, a viral ecology lab, kind of got into some of the protein work I'm going to tell you about. So we applied and were awarded a grant through the National Science Foundation's Big 10 Ideas. And that was titled, "Does Replicon Biochemistry Define Infection Dynamics of Viruses Within Ecosystems?" Excuse me. Let me slow down a minute. Right. So you can tell kind of just by that title that we are still interested in the ecology of viruses, but moreover, we're interested in the biochemistry of the enzymes that those viruses encode. To accomplish the protein biochemistry work, we made this a collaborative proposal with some industry partners that I ended up doing a 2-week rotation with to learn all things about proteins. And that collaboration also came with the JGI sequencing grant awarded to Dr. David Mead that allowed us to get our construct synthesized. So those industry partners are from a company called Verogen Biosciences in Madison, Wisconsin. And the 2 people that I worked most closely with are Drs. David Mead and Phil Brum, who are the CEO and VP respectively. Verogen's mission statement is attached here, but in short, they're interested in using microbial enzymes for biotechnology applications, such as making new kits and discovering enzymes that have novel capabilities that can be used in molecular biology techniques. And these 2 guys supervised my rotation in Madison and taught me way more than I have time to tell you, but I will show you some of the data that we just recently published. That brings me to my research questions. The overall purpose is to connect the biochemistry of pol A to the phylogenetic variation that we see in the environment. Some of the leading questions include, can we actually have metagenomic sequences produce soluble proteins that we can work with in the lab? Are they active? Next, does that 762 amino acid position I told you about impact any of the speed or the processivity like we hypothesized from those historical mutagenesis studies? Are there any novel functions within these polymerases that could be good for biotech applications? Obviously, Verogen's very interested in getting biotech applications out of this. How diverse is the biochemistry across global diversity? And lastly, my little pet project piece, do the associations of helicases reflect biochemistry of the polymerase? Okay. So this slide shows you how we kind of started to answer those questions. It's yet another phylogenetic tree colored just the same as all the other ones, except this time we added over 2,000 sequences. We have a handful of bacterial sequences down here in gray used to root the tree. But by contrast, everything else in this tree is viral. So that really tells you the diversity of enzymes that we're working with. You can see once again, we have distinct groups by color according to the 762 residue. Even after the addition of thousands more sequences, these still hold up really well. And as I said before, it's just one amino acid out of 900. We think that potentially running into some confusion here with the phenylalanine groupings, there is some mixing going on. We think that that might be ancestral and basal to the tree, which is why we get some switching back and forth. So for this functional metagenomics study, we assembled and identified environmental POLA sequences from our metagenomes and selected 50 diverse viral POLA sequences, including some of the known reference phages over here, as they do already have infection dynamics that are published so that we could leverage that. So these genes were codon optimized for E. coli, synthesized by JGI, cloned into E. coli cells, and then we got some proteins that we could work with in the lab. So this slide shows you STS page gel of our protein products from a subset of those selected sequences. The naming conventions here are if it has an R in the name, that's a reference page, so a genome sequenced reference page. And if it has an S, that means it's derived from a viral metagenome. So all of the sequences here were synthesized with a 6X his tag on the N terminus so that we could purify with a nickel column. Now this is all post-nickel column, so these are our final protein stocks. But as you can see, so the red boxes here indicate the protein bands of interest between 64 and 75 kilodaltons. And it's very, very clear from this gel that these are not the purest stocks in the world. And I will be the first person to admit that. We do have a lot of troubleshooting to do with this, but the good news is that we did have a success rate of predicting POLA sequences from viral metagenomes and producing soluble proteins of about 60%, which I think is pretty awesome. And the even better news is that even though they're not the highest purity, they are actually active. So this is an agarose gel showing some representatives in a primer extension assay. The top clone is FS24 and the bottom is FS21, so both phenylalanine proteins from our viral metagenome. And this is showing you that we actually did this from metagenome assembled sequences, which I think is really awesome. We tested the functionality of the polymerase on 2 different substrates. So the left here is showing you single-stranded M13 DNA, and the right is showing double-stranded M13 DNA. And the first thing that we can see that I have pointed out here is primer extension. And you can see by the shift up in the band, sorry, I should say the triangles here are showing you decreasing enzyme concentration in the reaction. But as compared to the negative, our enzymatic reaction, the shift up from single-stranded M13 shows you that the polymerase was actually able to replicate that entire single-stranded piece. So you have a band 2 times the size of the negative. And then the next activity shown is strand displacement, and this is exemplified by our positive control. We used 529 as a positive control, which is a famous strand displacing polymerase used in a lot of biotech applications. And you can see that the big streak upward here is showing you that giant molecular weight piece of DNA. And we see that in our test enzymes that we do have strand displacement activity at high concentrations, but not so much at the lower concentrations. An interesting point that we discussed at length in the manuscript is, why would this biochemical ability be advantageous to a phage? Granted that in vitro does not always equal in vivo, but maybe the phages that carry a polymerase that strand displaces are in need of one because they don't have a helicase. But I'll talk more about that in a moment. So to summarize the data that we have to date and what we put in the manuscript, I've got yet another tree, same branch coloring that you're used to, so I won't belabor that again, but I do want to let you know that this contains all of our synthesized sequences as well as some characterized representatives, references like E. coli, TAC, and T7. The next 2 bars here are showing you the source of the clone and the environment of origin. So the source, by that I mean if it's from a genome sequenced reference phage, a microbial metagenome, or a viral metagenome. And the difference there is just how the sample is processed. And I can talk more about that later if people have questions. And then the environment of origin is a little bit different. So the categories here are estuary, a few different hot spring environments, marine, or unspecified. And unspecified just applies to things like E. coli because it's everywhere. The marine environment is where some of our reference pages come from. And then the estuary is the viral metagenome that we used. And then the hot spring environments are polymerases that were selected by Verigen for biotechnology hopes. They were looking for a thermostable polymerase, which is why they selected from hot springs. And we did get some very interesting biochemistry out of these guys. So the next 3 columns here are showing you that biochemical activity for each synthesized pole. The light gray is showing you whether or not the protein was produced. Presence of a bar is yes, absence of a bar is no. Dark gray next shows you if that protein had primer extension activity. Then the black shows you if that protein had strand displacement activity. And these last 2 columns here, the dark gray and the black, the length of the bar denotes the strength of that activity. And interestingly, we do see strand displacement activity across a pretty diverse phylogeny of pole enzymes. But the key thing here is it's limited to only phenylalanine and just one leucine, 762 residue. All of the tyrosine polymerases that we tested did not show strand displacement activity, suggesting that maybe the increase of speed may not allow for strand displacement to occur. Additionally, given that only one leucine 762 polymerase had strand displacement activity, and it is quite closely related to the phenylalanines, that maybe the increase in fidelity given by the leucine residue also limits strand displacement activity. And lastly, on this figure, are colored bars indicating the adjacent helicase type to the polymerase if applicable. The light green here is for GP4, and you can see that these are all concentrated up here with our tyrosine polymerases. The blue then denotes SNF2, which is that non-helicase I told you about earlier. And then the tan is any other type of helicase, whether that's DNA B, RecD, or something else. What I'm really trying to show here is how different the associations are between the tyrosine polase and the others. Almost always when we see a tyrosine pole, it has a GP4, but we never see a GP4 with any of the other 2 residues. And most of the blue and tan bars are with the tyrosine or the leucine and the phenylalanine. So additionally, this SNF2 non-helicase occurs most often with leucine, suggesting, it moves more slowly. Maybe it doesn't need a helicase that actually separates strands. But nonetheless, 6 of those 7 strand displacing polymerases either encode an SNF2 or don't have any identifiable helicase within the contig or the genome. So just trying to represent this a little bit more friendly to the eye. Here we have a grade of enzymatic speed, and you'll see the polymerases fall along the spectrum, tyrosine being the fastest, phenylalanine being in the middle, and leucine being on the slower side. And those are according to those mutagenesis studies from the 80s and 90s I talked about earlier. I've also added R&R here that I mentioned a little while ago, and you'll see that it's only occurring with tyrosine and phenylalanine. And same kind of thing with GP4. We only ever see it with the tyrosine 762 pole. And the other 4 bubbles here are the different types of helicases, SNF2 occurring with leucine and phenylalanine, and the other 3 kind of shared with throughout. So I'd just like to wrap it up here by touching on some points of the discussion that we recently published. Firstly, the use of this functional metagenomics approach as a tool for genome to phenome studies in viruses of microbes. I'd say that we were pretty successful in this. As I mentioned, 60% of the sequences that we selected from metagenomes actually produced soluble and active DNA polymerases. Next, the genome associations in pole biochemistry. I just kind of talked about that. The study added additional evidence to our previously developed hypotheses about helicase type and R&R association, depending on that pole 762 residue. And last, selection pressures. The strict selection pressure that viral genomes are under may be fueling this biochemical diversity of viral DNA polymerases, especially given the positive correlation between strand displacing polymerases and SNF2 family enzymes. So just to conclude, the level of success that we've had so far really lends promise to this approach for genome to phenome studies of environmental viruses. It's a pretty new application of functional metagenomics, and it's really exciting to synthesize sequences only ever predicted and observed from metagenome assemblies and test that biochemistry of the proteins that they encode. So as I said, we had about a 60% success rate for protein production. The proteins that did produce were 76% of them were active and did primer extension. Then 30% of those active proteins also had strand displacement activity. So this is a big step in the direction of phenotypic data collection. And we hope to add some of the metadata from the study to our global POLE phylogenies to better predict virus and viral infection phenotypes from the more readily available sequence data. So I have a lot of folks to thank, starting with the 3 advisors from the Vail Lab, as well as our collaborators in this work, both at Verogen Biosciences and NC State University. Dr. Jacob Dooms, who was our postdoc before he moved to NC State, has been really, really helpful in this work. And the guys at Verogen have been awesome with all the finicky protein work. Most of my undergrad I spent working on DNA level, so learned a lot in just the last year working with them. Additionally, my committee members, as well as all my lab members and my funding through NSF and JGI. So with that, I'll take any questions. And if you want to read more about it, I've got the QR code for our publication here.
Any questions for Rachel?
Just a very general question, like the residue 762. So do you know if there's any correlation between the residues there and the host that the phage infected?
If there's any correlation between the residue the virus carries and the host that it infects, I don't know that anybody has looked at that, but that's a really good question. It'd be really interesting to see if that is, if we can find a correlation there. I don't know that anybody's looked at it. I haven't.
Also, like people had talking a lot about using phage as a antibiotics alternative. Do you think that it would really help? Or what is the drawback of using phage as the antimicrobial treatment?
What's the drawback of using phage as antimicrobial treatments? I'll be honest, I haven't really read much about phage therapy. I do know that my PI, I think, was looking to apply for a grant on it a few years ago. I don't know whatever came of that. But potentially a drawback of using phage for antimicrobial therapy is, if you don't know all the genes that are encoded by the phage, there could be something that causes, pathogenicity of the host that it infects if it integrates into it. That could be a potential drawback.
Thank you.
Are there any ways to match the phage sequence with host?
Any ways to match phage sequence
Yeah, so
with
can you
host?
predict the host from the phage sequence?
I'm not sure. I'll be honest, I'm not sure. Our typical uses of viral metagenome data, when we sample, we try to do a microbial metagenome and a viral metagenome. In my time in the lab, we're working on getting both of those sequenced at the same time. We'll do the 16S analysis on the microbial metagenome and some of the marker gene studies on the viral metagenome, but I don't know that we've gotten really far enough to get to that sort of question of predicting what host an environmental virus infects.
Just to comment, you can come at from the other direction. If you've got the genome sequences from the metagenome of the hosts, you'll sometimes get signatures in the CRISPR-Cas repeats, which will be homologous to your viruses and can tell you that that virus has once infected that host.
Right, yeah.
One of the ways of doing it.
Now that you mention it that way, there was a student that graduated with his PhD, I think a year ago or so, who looked at mapping to try to predict prophage regions within his bradyrhizobium culture collection. So that is one way to look at it.
It's a long way to go.
Yeah.
Are there any other questions? I'll ask one. So you had shown that the different residues at 762 seem to be associated with different helicases.
Mhm.
So is the hypothesis that you had to change the DNA polymerase first and the helicase changed afterwards, or vice versa?
I would say the polymerase probably has to come first, because it's the main enzyme that's used in DNA replication. And a lot of times, I think in T7, especially, that tyrosine, pol A, and the GP4 have actually been shown to interact at the replication fork. But yeah, if you don't have the polymerase first, it's kind of tough to evolve a helicase that it needs, I would think.
Yeah, in that case, if there's no more questions, thank you, Rachel.
Thank you.
Today is Alexander Wenzel, who is from Syntec Industries, and his talk will be on new natural products from marine cyanobacteria and microbiomes.
Yeah, thanks a lot for the kind introduction. Yeah, I just want to give you an update about where we are with respect to our toolbox development on finding new natural products from microbes, be it isolates or microbiomes. And as you know, we've been working with marine bioprospecting for a long time, 20 years about, approaching different environments, marine environments, including oil reservoirs offshore, which we still consider also a marine environment, and trying to find, for example, new bioactive compounds, new enzymes, but also other products that could be valuable in a commercial setting. For those of us who do not know where we are located, this is we are located in Trondheim, Norway. This is the Trondheim fjord, which is our local fjord, where we are focusing a lot on. So you see, it's Norway's third longest fjord, and most people say it's also Norway's most boring fjord because it doesn't have the large and spectacular mountains on both sides like the two others. But still, it's a very interesting place. It has a lot of freshwater influxes. It has diverse marine niches, and it's very deep. And as you see here, this is Trondheim. You see the fjord in the back. And you see we are located at 63 north, which is around like the southern part of Baffin Island, if you look into this on the Canadian map. So what is interesting, we have, for example, a coral reef, a cold water coral reef in the fjord, which is probably a pretty unique place to go. And there's a lot of sponges in there as well. I'm coming back to those sponges during my talk. So we've been working with a lot of marine actinobacteria, isolated from diverse samples from the Trondheim fjord. And we have built a strain collection that Sean had already mentioned of about 8,000 isolates, actinobacteria isolates, with a very large diversity and a focus on rare actinobacteria. So we've been applying the classical approaches of bioprospecting, finding new bioactive compounds for a long time. And so there has been some successes in finding new chemistry from those samples. So of course, so we want to go beyond that. And of course, what's important is to focus those natural products that are really novel. So reducing the rediscovery rate is a really big issue, and there's different things to do that. Of course, going towards environments that are not studied that well, but also looking into those microbes and those biosynthetic gene clusters that are cultivated, where we do not see an expression of these biosynthetic gene clusters, no formation of that chemistry that is encoded in those. In addition to, of course, looking into metagenomics, which is kind of a focus also of the conference here, to look into those non-cultivable fractions. So what you need, of course, is a toolbox for prioritization, like bioinformatics workflow, and also the technology to put this into action in the lab, which is the BGC cloning, the transfer into expression hosts that work with those clusters. Of course, you can optimize or at least assess those hosts and develop those hosts to give them a good prerequisite for actually expressing these BGCs and producing the compounds that are encoded. And of course, you should ideally do this in a quick way. This is why we're also looking into high throughput bioactivity screening, structural elucidation or prediction. And lately, we've also been looking into the possibility to pre-enrich certain fractions to get the diversity down into a direction that we want to go, and apply new cultivation methods to get more of those strains of this bacteria that are uncultivable actually in the lab, cultivate them, and to get the BGCs, get access to the chemistry that is encoded in the BGCs of those. So we have been building this platform, which we call InBiofarm platform, because it was developed in a recently concluded project called InBiofarm from the Center for Digital Life Norway, financed by the Research Council of Norway. We have a lot of international collaborators. So I will just give you a few updates about the different components of this platform, which is a bioinformatics prediction of the compounds encoded in biosynthetic gene clusters, the gene cluster cloning and expression host transfer technology, the development of the respective hosts, where we are using systems biology approaches, and finally, the compound identification and bioactivity assessments. And all these things together are built based on that marine Actinobacteria strain collection. But what is important to mention also in this context is that our aim is, of course, to look into, to open the possibilities to using BGCs that are derived from environmental metagenomes as well, so that the technology that we are generating is generic to that extent as possible to also assess BGCs from uncultivable ones. So the first part is, of course, we were looking into the diversity we have in this marine Actinobacteria strain collection. So we had a great collaboration with Mark Lyles, Auburn University, where we looked into the 1,200 draft genome sequences that we have of selected isolates from that strain collection, and make a phylogenetic tree to see, okay, where is the differences, what clades are new, and can we find this? So he applied a tree generation based on housekeeping genes in relation to all at that point known Actinobacteria that were sequenced. So that was kind of mapped together, and we got a really good overview about like what we have. And of course, we used this strain prioritization for, of course, we did some new type strain descriptions based on that, because we get to know here's really new clades. So let's describe one of those. We used those draft genomes here in this case for selecting strains for genome closing in order to get the complete BGCs, because fragmented genomes usually do not give that up to 100 kb contiguous sequence to actually assign or really define the boundaries of the BGC. But also we were looking into which strains might be possible to develop into new heterologous expression hosts, because we have, it's widely accepted that you need to have a wide panel of different host species to shuttle those BGCs into in order to get an heterologous expression. So the closer the BGC origin is to an expression host, would increase the chances to actually get the BGC expressed and the respective compound produced. So we worked about all these things. For the first point, we, together with Mark Lyles and Megan Sandoval-Powers and Stanislava Kralova, working in labs in Mark Lyles' lab. So we did 2 strain descriptions. I just want to briefly go through this, that we worked systematically also on these things. So we described the Streptomyces tardus from a sediment from the Trondheim fjord, and ranked this to all known Streptomyces at this point. Of course, we had the genome sequence closed for those, and identified different BGCs that are available in this strain. And we found this candicidin-like BGC that has antifungal, that encodes for candicidin, which has antifungal activities. And we were able to confirm this also in cultivating that strain to produce candicidin. And you see here that there's a response from candicidin being produced in under this condition. But we also see that there's a peak here which corresponds to an activity that is against, for example, enterococci. And so there's a lot of things to do from continuing with this isolate in order to see if we can find some interesting antibiotic activities. The second example is we named Streptomyces puripherorum because it's derived from a sponge, and this is an isolate which has anti-MRSA activity. So that's why we selected it, in addition to the phylogenetic novelty. What is interesting when we look at the BGC content of that strain, you see it has a really large number of NRPSs and type 1 PKSs encoded that is completely sets the strain completely off those strains that are kind of surrounded from like phylogenetically surrounded to that particular strain. So this is we had a lot of predictions based on this diversity of BGCs that we have on the strain, and this is ongoing activity to further look into this. Okay, but of course we wanted to go large scale. I wanted to go towards our heterologous route to activate, for example, BGCs or express new BGCs. And so this is the bioinformatics workflow that were developed together with collaborators in Wageningen and Jacobs University in Bremen. And it's basically an integrated tool that includes different types of assessments of the sequences. There is anti-smash, which is of course very central in predicting BGC functions. And but we also complemented this with this BGC domain finder and TSNE clustering to maximize the BGC diversity, and also the Bigscape clustering in order to predict BGC novelty. So all in all, in that workflow, we ended up with about 20,000 BGCs predicted by anti-smash. And by integrating also the taxonomic strain novelty, we selected 88 of these 1,200 strains to look further into, because that seemed to be a more a diversity of little redundancy. So to really focus on something. So this is about 3,200 BGCs. That looked like this, and it's a lot of NRPSs, a lot of terpene clusters, and based on that overall assessment, we finally selected 35 BGCs, very diverse BGCs, including 7 model BGCs. So it's 28 actually from that set of 88 strains that we were specifically focusing on. And of course, now we have selected that. Now, of course, we need to get this into a form where we can actually put this into a heterologous host. So we need to clone that BGC. And you see here from this first 18 of those that we actually have transferred to heterologous hosts. So the others are still pending. You see that the range of sizes is up to 130 KB. And so these were those that we were most interested in, including mostly NRPS, PKS, but also some others. And also the models, the model BGCs that we selected from the respective hosts, normal bacteria that produce these compounds, also include a few antibiotics, including nystatin, which also has a 130 KB cluster. So it's a big challenge to get those cloned, but luckily we have that collaboration with Virogen Biosciences, who have developed a technology to cleave out BGCs, or large chunks of DNA from genomic DNA. And so we shipped those strains off to Virogen, And they used their technology, which is a Cas9-based restriction digest of the chromosomal DNA, very specific, that then leads to a fragment that can be combined with an expression vector. And this P-dual-P expression vector developed by Virogen has two inducible promoters on both sides of the integration site, and that allows an inducible expression of what is in between. Of course, we do not know, it's not very directed, so sometimes it works, sometimes it doesn't, but at least we often see effects, and in some cases it really happens, it's really a positive effect. So all these BGCs that we selected were cloned by Virogen. So this is the 6 model BGCs. Of course, it's tested by restriction digest according to the predictions. And we also have our novel BGCs, all of them cloned. So it's just really been a big help to have this technology in place in order to get hands on these complete BGCs in the form that we can transfer it into our expression host. And our main expression host is Streptomyces silicolor. For historical reasons, we have been working a lot with this strain, but of course, I mentioned earlier, we are also looking into alternative other strains in our host panel to be able to have a broad spectrum and then increase the chances of having a new compounds being produced. The systems biology approach, we were working a lot on together with collaborators in a European project to really get a genome scale metabolic model, Streptomyces silicolor, in order to be able to change it according to optimized expression of what is encoded. And so we were basically developing this SCOJAM community model, which is out on GitHub and is supposed to be developed jointly by the Streptomyces silicolor community to improve its predictabilities. As an add-on, we developed a pipeline which allows us to integrate BGCs in the genome scale metabolic model of Streptomyces silicolor. So this is a tool which we call Big Mac, which takes the BGC annotation file of the anti-smash and parses the information into domains, into modules, and tailoring genes, for example, in order to assemble a predicted pathway. And if we have that in that format, that we, for example, also have the different domains of polyketides and NRPSs included, we are able to integrate this, plug this into the framework of GEM, and then make a prediction about how that actually is working and what is needed with respect to precursors to produce that compound. So we, oops, a bit too fast. So we verified this the function of that pipeline by taking 8 biosynthetic gene clusters that are well annotated for the pathways, and our algorithm is able to predict about 75% of those functions automatically, and also by integrating this into the GEM of Streptomyces silicolor, we were able to look into predicted production rates, compare this with literature, and also look into gene knockouts. So if you want to knock out a gene and you see, okay, then you get a production increase, we also see a very good match of our predicted integrations versus what is reported in literature for the respecting BGCs. And then we did this at a large scale. So we selected 943 BGCs from MRBig, did the automatic annotation, so you see it's a very wide spectrum of different BGCs, different origins, NRPSs, PKSs as the focus, and again checked for production increase if we apply a certain knockout in our Streptomyces silicolor strain. So this is it doesn't really help a lot. So we get a few percentages. So there's still potential for improvements of this, but the concept is there. So plugging this in will give us the possibility to prepare the production host in a way that at least the precursor supply is not a limiting factor when you want to increase, want to have a BGC heterologously expressed in silica. So of course that is one aspect on the precursor supply. We also worked on of course the gene regulatory network, which is probably the main bottleneck in this sense, and also nitrogen metabolism. But a key that we work on is morphology engineering. Streptomyces silicolor is growing in clumps, in mycelia clumps, and that is a big problem when it comes to scaling production in actinobacteria in general, but of course in Streptomyces silicolor in particular. So we were using the John Innes Center M11 series, in particular the strain M1152, which has a kind of purified MS spectrum, and which worked most well in our hands. And what we did with collaborators in Leiden University is deleting a small locus, which is called MET-AB, which encodes for poly N-acetylglucosamine synthesis. And by deleting this, we were able, or the collaborators were able to basically dissolve these clumps, and so we get a mycelium that is much more open, and we actually get a very nice growth curves from this, which is not kind of comparable with the wild type, which is the red ones. We actually get an exponential phase until it runs into a limitation. So that's a very good thing. Of course, we deleted that locus, of course, M1152, which is our favorite strain, and then we reintroduced the actinorhodine and the red cluster, and this is the summary of what we see. First of all, we see what I mentioned, like if we take away the MET-AB, we see an increase in the growth rate. So this is reproducible with 1152. When we delete the MET-AB, we get a massive increase in growth rate. So if we then add the red cluster or the ACT cluster, we see this to some extent still, but not too much when we add the actinorhodine. And one reason for that is probably that what you see here. So we see a slight decrease in production in the wild type. Of course, the 1152 doesn't have the red and ACT cluster, so we don't see anything. But if we introduce the red and if we introduce the ACT, we still see the decrease in production rate and yield. But what we see is we get a massive deregulation of the production. So by taking out the BGC and reintroducing it into the 5C31 attachment site, we can basically deregulate the expression of the BGC, and this is something which is an interesting observation. So that brings me to a use case, because we were putting together the things on the different developments. And so I'll focus on one strain, and in particular one BGC from that strain that we selected. It's a strain which is related to a micromonospora, and that strain had shown biological activity against gram-negatives, gram-positives, and some gram-negatives. And the BGC number 16 showed a very low, similarity, but interesting similarity to monomycin. And so we're focusing on that BGC. The BGC was cloned by Varigen using that PDULP technology, and then we transferred this into M1152 MET-AB, the mutant which grows as the open mycelium. So, and we see two new peaks appearing here when we have the BGC included, and so that was promising, and this is why we focused a lot of our efforts on this one and not on the 17 others. And so we did some induction studies, because the PDULP is an inducible vector, so we can have the two different inducers. We didn't see a really big effect in that, but of course, at least it didn't go down. So we were working together. We were working with cultures that are not induced, because it didn't really really help a lot, but just want to show those data. And then we determined the mass of that compound that is under the peak, which is about 1,350. We did some cultivations on label substrate in order to close in on the molecular formula. So we have 2 Ns and 61 carbon atoms. And we did then an upscale production and purification of that compound and determined bioactivity. So what we can see about activity, we tested against gram positive and gram negative. It had an effect against gram, you see here we have around 1 microgram per milliliter mix 70 of our compound compared to vancomycin, in the range of vancomycin actually. And for the vancomycin resistant, we see that we have a good effect with our compound, while of course the vancomycin doesn't really work on this VRE strain. So it's a promising compound. It obviously has a different mode of action than the vancomycin. And we also studied the toxicity against human cell cultures, and we do not see any toxicity in that range that we have tested, which is quite a prerequisite in order to develop this further. So then we solved the structure together with Red Lead in Sweden, helped us with that. And so you see the monomycin to the right, and this is our compound. There are some, like the overall structure looks a little bit similar, but we have some really unique features. One is these 2 ring structures, which are up here. One is that this is a phosphoglycolipid. We don't have any phospho, so we cannot call this phospholipid. So we have an ether bond here, while here's a phosphate. And what we have a sulfate group, which is located either here or there. So we couldn't unravel this yet. But and we have also some other slight modifications other places. So it's an interesting new molecule with a structure which is new. And so that's why we termed this nidaromycin after the old name for Trondheim, which is Nidaros. So let's see where this gets us. So that's that use case. And since we have the Trondheim picture up there, I just want to have my last 2 slides because we have recently, just going back to the roots, 20 years ago, where we built up the strain collection, there was no metagenomics. Now we have been going back to the same places within this Marbles project. We were using our research vessel from the university, Goneros, and we went out to the fjord to the same places where we derived the sponges back in time. And we actually found the same sponges there. So we were lucky. So I had some thoughts about, okay, what happens if we do not find them anymore? But 20 years is not a long time, obviously. So we could still find them. So this is the 4 species that we were looking at. So we have been sampling them, and we are now working on a full-scale omics approach in order to see how these microbiomes associated with the sponges work. We want to apply new cultivability studies by mimicking sponge structures in the lab and trying to grow biofilms which resembles as good as possible the original habitat, and then work towards really at full omics scale to try to understand in order to find elicitors that both enable growth of those strains in the lab, but also the interactions and in order to be able to upscale this and try to find out if we have new BGCs that are produced that are used to produce new compounds, either in the microbiomes, but also applying a functional metagenomics approach by using some of the tools that we have in our platform. So that is in 2 years, hopefully we will have some more on that topic. And finally, I just want to thank collaborators and from the university. So my collaborators in Sintef from the university, Of course, the group from Mark Lyles and at Varigen had a really technological boost on the development of the platform. The bioinformaticians from Jacobs University and Marnix from Wageningen, the people from the Systract consortium doing on the GEM work in Rostock and in Chalmers, the MET-AB story together with Leiden. And what I haven't talked about is the nitrogen metabolism and some other efforts on precursor supply together with Tübingen, as well as the MARBLES consortium and a few collaborators that helped us with the structure and some sequence work. So with this, I would like to close and thank you for your attention.
Nice talk. Do we have any questions for Alexander?
Thank you, Alex, for this great presentation and great results on your BGC. I have two questions, in fact. The first one is regarding the control of the Q expression of each of the genes of your BGCs in your recombinant host. Can you control that?
Now, of course, and we haven't even studied that. So this is one of the things on the list, in particular for those where we do not immediately see a production of a new compound. So we need to see to which extent is the transcription actually limiting. And this is also something that we want to look into specifically in the MARBLES project now with these new isolates and new cultures. So what happens at the transcription level in the original situation in the sponge versus a situation that we mimic in the lab versus an isolate situation of one of those compounds. So this is going to be very interesting, but currently we don't have that possibility to control individual modules of the BGCs, but this is something that Varigen is also offering now, so they can drop in promoters. We have some other collaborators at the university and also some commercial ones in Norway who have technology in order to refactor these BGCs.
Okay, thank you. And the second question is because I'm very curious. You mentioned oligosaccharide production. In one of your slides, BGC targeting the synthesis of an oligosaccharide. Can you tell us what kind of oligosaccharide it is, and did you succeed in producing it?
You mean this one of the BGCs?
Yes.
That that is of course we could not follow up on. It was a little bit stressful at the end of the project, so we only focused on that nitromycin story. But of course, it's definitely worth it to look more into the 17 others, in addition to the 10 BGCs that we haven't even conjugated into streptomyces. So this is something. If this is something of high interest for you, it could be a possibility to collaborate and try to evaluate this together.
Except
Absolutely.
that we should.
But we don't have data on this right now.
Because we have to deal with transmembrane glycoside transferases, so it could be very difficult in recombinant hosts.
Yeah, one should have a look
Okay.
at the BGC and see what is actually encoded there.
Okay.
And yeah.
Thank you.
Any other questions?
Thank you for the talk. So I was very intrigued when you showed us the maths and how you dealt with the morphology. So I don't know much about like how similar for other fungus, for example, that have similar morphologies, which is a bottleneck for scale up in fermentation or bioengineering. Would something similar be done? Like, can you apply this to other fungi as well?
Yeah, so of course, this is like streptomyces is a bacterium, so, and this has been tested. The deletion of the MET-AB cluster shuts down the N-acetylglucosamine, poly-N-acetylglucosamine synthesis, and that leads to an open mycelium. So this is something which is transferable beyond Streptomyces silica. It's even more. You can actually try to take that and include this into another strain, which actually grows in open mycelium, and let it produce it. And it starts, in some cases, forming clumps. So this is how the collaborators actually showed that this is a general phenomenon. So if this cluster is present and expressed, you get clumps, and if you remove it, it's an open mycelium. Not 100%, because there might be other functions, but this is proven.
I was kind of curious about this GoGem because I do metabolic modeling of rhizobia. So I was curious how well that's working. Like, is the community coming together to help with that? And people that aren't like metabolic modelers, are they interested?
Yeah, of course, it's not a huge community, and of course you need to have the community actually adapting it. I haven't checked lately how many. So there have been some people going in, making suggestions, making changes, making new annotations. Everything is tracked on GitHub, but it's definitely not a. So we should promote it more. But there's also some competition going on, because we were taking different existing models and we're merging them into one and saying, this is the one where we can go further with. Of course, those people need to accept that. And yeah, so it's probably a little more work that needs to be done in order to really make people use it. But it's out there, and it has most reactions from all models so far. It should be a good starting point for further improvement.
Nice, thanks. If there's no more questions, thanks again for the talk.
Thank you.
Our talks from the poster abstracts. So the first two talks are members from the Open Plastic Project, which is a Genome Canada project here, well, in Canada. And it's a multidisciplinary project looking to develop biological solutions to the plastic problem. And so our first speaker from that area will be Jardin Lier-Hood, who will be telling us about his talk on covering the microbial capacity for plastic biodegradation.
Hello, does this work? Okay. Hi, everyone. I'm Jardin. I'm a member of the Trevor Charles's lab, and also being co-advised by Laura Hug, who's not here but wanted to come. So just a disclaimer before I start, I don't really have a lot of primary results to share with you. This is more of a lit review and just a current state of the industry with my future goals. So without further ado, plastics are ubiquitous materials in today's society with broad applications in many industries, and their ubiquity stems from their desirable physiochemical properties and their low manufacturing costs. Unfortunately, these same physiochemical properties render them extremely recalcitrant to natural biodegradation processes, which are employed by microbes and their enzymes. Thus, they persist for decades to centuries. So globally, in 2020, there was 386 million tons of plastics produced, which is equivalent to the weight of 92 million elephants. And this is expected to increase by 8.5% annually. 40% of these plastics have single-use purposes, which is a stark contrast in longevity versus service life. Currently, only 9% of plastics are recycled, with the rest being processed via landfilling, incineration, and dumping into the ocean. So currently we live in a linear use economy where we take carbon from the earth, we use it to make plastics, we use the plastics, and then we dispose of them. Where what we should be doing is diverting this plastic waste using novel biodegradation technologies, essentially recycling it into the system generating more of a circular use economy. So here is a proposed circular use economy, a novel biodegradation technology proposed by Rue and Associates from their 2020 paper. So essentially we take plastic mixtures, we break them down into smaller fragments to use as a feedstock for biological depolymerization. From here, we can take these depolymerization products and use them to make more plastics, or we can also use them to make all sorts of products. I have a pointer here. Sorry, I don't know how to use this. Oh, top one. Biodegradable plastics, intermediates for chemical reactions, and products of added value, such as biosurfactants. So essentially just valorizing plastic waste. So the two plastics that I am interested in are polyethylene and polystyrene. So polyethylene, the one on the left, is a fully saturated hydrocarbon, essentially an N-alkane. And then polystyrene on the right is has a similar structure, but it has alternating phenyl groups. So we can examine plastic recalcitrants on multiple levels. On a primary level of the structure, these are hydrophobic and non-hydrolyzable molecules. On a secondary level, they have high molecular weights, and degrees of branching can affect biodegradability due to steric hindrance. And finally, on a tertiary level, the degree of crystallinity really impacts biodegradability due to these high crystalline regions being tightly packed and not accessible to enzymes. And then finally, surface area is a factor to consider when thinking about enzymatic reactions. So here is the current consensus model for biodegradation of plastics. It has 4 steps. Oh, okay. I don't know what happened there. Okay, so step 1 is deterioration. So these plastics are, these are virgin plastics, which are non-hydrolyzable, thus requiring the use of extracellular enzymes or abiotic factors to essentially oxidize the surface level or modify the surface level, usually oxidation. Once oxidized, enzymes can hydrolyze these modified functional groups. I don't know what I'm doing here. Keeps changing. Touching something. Essentially generating shorter chain fragments, so oligomers and sometimes monomers of various oxidation levels, which can then be assimilated into the cell, the 3rd step. And then finally, these are broken down within the cell, so fully mineralized, using catabolic or anabolic processes. So what is currently known about microbes that break down plastics? So this data has been generated from the PMBD, the Plastic Microbial Biodegradation Database, and essentially shows microbes at the genus rank and the number of times they've been reported in studies. So you may notice that for polyethylene on the left here, it's truncated and there's 30 more rows. So there's a lot more research that has been done on polyethylene, whereas polystyrene only has 9 known microbes at the genus rank. And they're both dominated by Bacillus, followed by Pseudomonas, and there are some fungi here as well. It's worth mentioning that at this point, there is some studies that have shown that the gut microbiota of insect larvae are able to break down polyethylene and polystyrene at a much faster rate than just the bacteria on their own, or the microbes on their own. So a lot of these studies have been culture dependent, where we sample from anthropogenic environments, we isolate microbes, and then we characterize enzymes. The limitations of these studies are that they do not consider cases where biodegradation is a cooperative effort within a community of microorganisms. So for example, within the gut microbiota of an insect, there may be several key players involved. Additionally, the vast majority of microbes are not amenable to culturing, so we can't actually get them onto a Petri plate. So essentially, the system is greater than the sum of its parts. So to address these, we use culture, or potentially to address these, we use culture independent methods. First one being metagenomics, everyone's familiar with, where we are trying to isolate metagenomic DNA to construct libraries to express in suitable hosts. The host that I will probably be using is Pseudomonas KT2440, which is a very well-described, well-behaved domesticated bacteria, and is highly suitable for complementing with functions of interest. So then we grow this on selective plates. In this case, minimal plates where the sole carbon source is plastic or the monomers of plastic. This way we can detect biodegradation and utilization. We can complement this with sequence-based metagenomics, which essentially requires reference sets of chemically validated enzymes. We then use these to generate hidden Markov model profiles, which essentially capture the variability within consensus and non-consensus regions. So similar to BLAST, but more sensitive. And then we can use these profiles to query global shotgun metagenomes, thus to predict putative orthologs. So this method is highly dependent on having chemically validated reference sets, essentially garbage in, garbage out, right? So what do we currently know about polyethylene and polystyrene enzymes? So again, this has been generated from PMBD, and it shows a number, or it shows enzymes that are suggested to be involved in the biodegradation of polyethylene and polystyrene. So notice you can count the number of enzymes on both hands, and there's actually only 2 known enzymes for polystyrene down here. And these are not actually biochemically verified. These are suggested to be involved in biodegradation. Only one of these actually has a uniprot accession. So currently we do not have viable enzyme options to use as profiles. So this is essentially a proposed procedure for my project. We would like to be able to do computational screening of global environments, but we don't have input sequences. So we will perform functional screening on high potential environments such as insect gut microbiota, wastewater affected by plastics, and soil dump sites, for example, to potentially, or hopefully, I guess the project really hinges on identifying active enzymes to use as input sequences, which then after each iteration, we can hopefully generate more and more enzymes. So the expected output of this project is an initial library of polyethylene and polystyrene degrading enzymes, which can be used, which can be used as the foundation for industrial scale up, and thus pivoting from a linear use case economy to a circular use case economy. Additionally, surveying global public metagenomic databases will allow us to assess the distribution of these enzymes across the microbial tree of life and within global environments, thus highlighting regions in most need of intervention. So I just want to thank the Open Plastic Project for giving me the opportunity to do this research, and Genome Canada for funding us, as well as the sponsors of this conference. Thank you.
Any questions?
Great talk, thanks.
Thank you.
I was wondering, what do you think about, yeah, I would expect that there are several different enzyme classes that are necessary in order to see a degradation. How do you approach that? By combining different functions, by holding one function constant and screening for the next one, or?
Yeah, we were thinking of starting from, I guess, starting backwards, where we would look for enzymes that break down the monomers. So polyethylene is an N-alkane, so we could use some sort of alkane structure, longer the better, I guess. And then screen for enzymes that break down that, and then hopefully they would have promiscuity towards larger polyethylene fragments. And then for polystyrene, we can use the monomer styrene. Although it's not exactly the same shape as polystyrene, there are other dimers of polystyrene, but yeah, essentially working backwards. And then at that point, if we can complement Pseudomonas petita KT2440 with the function of being able to utilize the monomers, then hopefully we can find additional enzymes that can perform the critical biodegradation steps, so the oxidation and hydrolyzation. So yeah, working backwards.
That's good. Thank you.
I mean, the complexity here, of course, is it's not a single step process.
Yeah,
It's a multi-step process. I mean, the enzymes you listed there, they're almost exclusively activation enzymes.
Yeah.
Putting in a reactive group such that you can then get a secondary cleavage and breakdown. So it's a tough call.
It's high risk, high reward, I would say.
Yes. Thank you. Yeah, here. Thank you for this nice talk. You already performed a very nice literature review. And did you see any paper on growing metagenomic clones on mineral medium supplemented with polystyrene as soil carbon source? Did you already see any success using this strategy, or?
I've seen more success with the monomers.
Yes.
So C16, I think that's dodecane. It is the largest monomer before they turn to solids at room temperature or ambient temperature. So a lot of people have used that. But I've also seen Petri plates where they use suspensions of plastic particles. But the monomers would have a higher chance of getting hits from screening, I would think.
Okay.
I would assume.
Thank you.
Yep.
Thank you again for the talk.
Thank you.
Are talking about finding plastic degrading microbes and enzymes from the guts of beetle larvae.
I'll just stick with the laptop. Can everybody hear me? Okay. All right. Find the pointer. Okay. Thank you for to the organizers for giving me a chance to showcase my research. So I'm part of the same open plastic project, and Jordan did a really good job of introducing and giving the literature review. So I won't spend that much time on it. Yeah, my project, I guess the title is Finding Plastic Degrading Microbes and Enzymes from the Gut of Beetle Larvae. So just to give you stats, Canadians throw 3 million tons of plastic in the waste every year, and only 9% of it is recycled, and most of it ends up in landfills. And as Jordan mentioned, we want to create a cyclic economy where instead of the plastic waste ending in the landfill, we want to bring it back and reuse it. So there are 7 different types of plastics that we use in our everyday life, and they range, the decomposition of these plastics in the natural environments can range from few years to hundreds of years, and some of them never. And the recycling rates are also pretty low. So in our project, we are mainly focusing on the polyethylene and polystyrene, but we are planning on including PVC in our project as well. So these ones are especially difficult because of the homoatomic structure in them. The carbon-carbon bonds are difficult to break down. So This is a picture of beetle larvae. So we are currently working with mealworms and superworms, and these are used for human consumption or pet food as well. So we buy our larvae from a local pet store, and as you can see on the picture up left here, beetle larvae, like superworms, are eating polystyrene, and has been well established in the literature that they can eat polystyrene. And the bottom we see polyethylene. So we just cut our regular grocery bags into smaller pieces, feed them to the larvae as the only food source. And you can see the serrated edges. So they are chewing at those polyethylene pieces. And on the right here, we have a picture of dissected gut. So we take the gut out of the larvae after 10 days or so of them feeding just on either polyethylene or polystyrene. And the red arrow is pointing at the pieces of microplastic. So these white pieces here are polystyrene pieces, they're present inside the gut. So there have been different studies that published. Jordan also mentioned that people are trying to see if these worms can be used as a tool to degrade plastic, or at least this will be, because they're known to eat plastic, so this will be a good place to look for those plastic degrading microbes and enzymes. But our project is not just limited to that. We are planning on looking at wastewater samples, landfill samples, and manure as well. But the one thing that lacks in these studies is a concrete proof of, is this plastic is being actually degraded, or is just being converted into micro and nanoplastics. And the exact mechanism of degradation is also unknown. So us with our collaborators are trying to achieve that goal. So the objective of this project is to identify enzymes and microbes that have the potential to degrade plastics. And we are starting with a metagenomic analysis. In literature, there is no metagenomic data set present from any beetle gut. So this will be a good data set to have around as well. So just a timeline of our project. So we buy, as I mentioned, we buy our worms from a local pet store, and then we feed them on oatmeal and bran diet for 2 days to just normalize their gut. And then we put them on special diets for 10 days. So our diet treatments are polyethylene, polystyrene, or our standard diet of oatmeal and bran, and then we have a starvation control in there as well. So they eat their diets for 10 days, and they seem to survive just fine even without food for 10 days. And after 10 days, we dissect our worms and take the gut out, and then kind of squeeze it to take out the gut contents and use that to isolate microbial genomic DNA. For each treatment, in our current analysis, we dissected 30 mealworms and pooled their gut contents. And then for superworms, because they're slightly larger, we used 10 worms. So we isolated the microbial genomic DNA, sent it off for sequencing at Genome Quebec, and we got the Illumina pair and read sequencing with 150 base pair length. And once we got our data back, we performed a metagenome analysis, assembly and binning, and assembled the metagenome assembled genomes, or MAGs. After that, we looked at the abundance of these MAGs in various diet treatments, and also from the annotation of these MAGs. And we are interested in the enzyme. So the proteins of interest, we want to look at this data set and see if we could find, the assumption is there is a more likelihood of finding these proteins in this environment. Just to briefly give an overview of the quality of the MAGs we got. So we used 51 marker gene as our threshold, and select the MAGs that had over 70% completeness. So for mealworms, we had 53 MAGs after dereplication that showed over 70% completeness, and 10% were 100% complete, 79% showed over 90% completeness, and 11% showed over 70% completeness. Superworm samples were pretty similar. We had 100 mags from the superworm samples. We had 14% that were 100% complete, 77% over 90%, so and 9% over 70. So we got a good data set on our hands. So we performed phylogenetic analysis on these all 153 mags, and just looking at the diversity of microbes, we found we have a lot of firmicutes shown in the blue here. We have proteobacteria, gamma and alpha proteobacteria, and actinomycetes as well. So this tree shows this taxonomic level at the level of class, and the tree is rooted at midpoint. So the next part was the part where we were really interested in is does the diet have an effect on the abundance of the mags? And the brief answer we got is not at this point. So as you can see them clustering all together at one point. So we performed a PCA analysis, and you can see all 4 diets: control, plastic bags, standard, and styrofoam. And we don't see any small groups scattered on this PCA. So we wanted to look at the mags in each treatment. So this is a heavy graph, so I'll just walk you over. So the relative abundance is on our y-axis, and we have all the treatments, all the replicates shown on our x-axis. And the reason why I did that and not took the averages is because we see a lot of variation between our replicates. And one thing I didn't mention earlier is each biological replicate was isolated on a separate day. And initially we are seeing that the day of isolation does have an effect on the abundance we're seeing. Just to point out, so each diet has 3 biological replicates, 1, 2, and 3. And then from each biological replicate, we have 2 semi-biological replicates. So we grew our containers in we grew our worms in one container and took out worms on 2 separate days. So those are our semi-biological replicates, and they seem to have an effect. I'll just point out a couple of them here that we are seeing some trends in. So these blue bars we see, so these are spiroplasma or firmicutes. We are seeing them more abundant in the plastic diet. In most of the cases, there are a few exceptions, and there's only 1 or 2 that in our standard or control conditions that have this particular subset abundant, but in general, this is more abundant in the plastic diets. And then we have another one, Lactococcus jarvae, which also seem to be more abundant in the plastic diet. So similarly, in superworms, we see a similar trend. There is nothing that is jumping out, but if you look closely at the data, there are some that are more abundant in the plastic diet, for example, Citrobacter. So we just analyzed this data 2 weeks ago. So the next plan is to pay close attention to these mags. So the future work involves investigation of these genomes, the few mags that we will shortlist from our data, and also make a non-redundant data set from the proteins of interest from all the metagenomes, and then look for enzymes that might have some function related to plastic degradation. For example, we would be looking for proteins with signal peptides or lactases or hydroxylases, esterases. So the enzymes, as Jordan showed in his presentation, that have been suggested to have something to do with plastic degradation. With that, this is our team. George, of course, our PI. Sydney, she is our master's student working on this project. And then Jeremy here, he's our summer student, and myself, we are part of the DiCenzo lab, not the entire DiCenzo lab. And lastly, our collaborators and funding partners. And thank you all for listening.
Hair is much longer since that photo was taken. Any questions?
You're looking at the numbers of mags from the different growth categories. You're making an assumption there, which might not be valid, which is an increase in a certain taxon on a particular diet represents organisms that are actually degrading the substrate. I'm not convinced that assumption is necessarily valid. It might be organisms that are benefiting from the degradation of the substrate, as opposed to actually actively degrading it.
That is a fair point. And I guess it's just a starting point for us. This project was started in last August, so it's under 1 year old at this point. But we're trying to figure out, because our first thought was that we will see nice groupings and we'll go there and like shortlist microbes, but that's not the case. So at this point, we are trying to revise our strategies. So you do make a valid point. And I guess one thing that's not shown here, sorry, did I do something wrong? Okay. The other part of this project that we are also doing is trying to enrich and trying to isolate microbes directly. So we're taking the gut contents and putting them in minimal medium. There was a question on the previous presentation, and I would kind of answer that. So we are taking minimal medium, putting the plastic pieces in that and trying to enrich communities and trying to see if we have any microbes there. And we have this data set, and then we can always come back to this data set for those communities.
Yeah, I'm just adding to that. Of course, once you've annotated your MAG sequences, you are going to get hints, obviously.
Yes.
If there are a whole bunch of hydroxylases or the other oxidative activation enzymes in there, then it'll be a strong hint that you're looking in the right place.
Yes. So we will be identifying those proteins and trying to trace them back and see in the MAGs if we have those. Yes.
Just a general question. I know nothing about the lifestyle of the worm, but I just wonder why do they eat those kind of things? Like, is the microbiome max? All right, it's like I don't know what is the another way around. So like they have to eat that, and then the microbiome have to adapt to the food that the worm eat, or it's another way around. Like somehow the microbiome makes the worm eat those materials.
I don't know if microbiome is the driving, like what their microbiome is the driving factor in their choice of food. We didn't give them the choice, but they have been known in natural environments to eat plastics for many, many years. And I think maybe just for survival, and they don't seem to mind eating plastic that much, at least for the time we are putting them on this diet for 10 days, and even longer. They seem to survive well. They do lose some of their weight, and we see less septum in the plastic diets as compared to the standard diets. But yeah, I don't know why this choice though.
So how do we make sure that it's the plastic, but because I think the plastic that we use, they have many additives in there and many other type of compounds. So how do we show that they degrade plastic, but not other type of additives or some other compounds in the materials?
Could you repeat the last part, please?
The materials that make up of the plastic is not only the plastic itself, but I think also some kind of additives to make the material, I don't know, more stable or more flexible.
Yeah.
And I just wonder, like, do they digest those compounds, or do they really digest the plastic?
Yeah, I guess that's one of our questions as well, because a lot of papers, they have claimed that these worms can eat and degrade plastic, but we don't know if that's the case, or if it's just passing through them and just being converted into microplastics. And at least for our experiment, initially when we are feeding them, we are just using grocery bags, but they're not, I guess, it's not the pure form that we bought from a company. We do that for our microbe isolation. So, but generally those are clean bags, and I don't know about the additives, how those are behaving, like the ones that are already present in the plastic.
Thank you.
Yes.
Are you sure 10 days is enough to significantly alter the gut microbiome? Maybe it should be 100 days, except they'd probably die of starvation.
Yeah, we had to find a sweet spot. I was not present when George and Sydney did initial pilot studies, and they landed on 10 days. But we have let the worms go for longer on the plastic diets. And because we also have to, because they convert into the next stage in their life is pupa. And if we let them go on for too long, they just become those mummies, and then they're not that useful to us.
Great.
I was happy to see that spiroplasma was there, because I read a paper about my community analysis on mealworms, I think, and they were the main group there too. So it's consistent. I was just wondering, have you thought about using co-diets where you feed them plastic and maybe bran? But I've seen that actually decreases the mortality rate and increases plastic degradation.
Yes, so I have seen some papers where they have these co-diets where they put bran in addition to a plastic, or mix different types of plastics. For this, at least data set, we didn't do that. We just wanted to see what happens, but it could be something in the future that we might want to consider. And the metagenomic part is just one component. The microbial isolation is another component, and it would be something to consider. Some people have used starch as well in combination with plastics.
Yep. Thank you.
We're going to have to stop there. Thanks again for the talk, and we'll move on. There will be Eugenia Datzi, who will be talking about biocatalytic production of polyhydroxyl alkanates using petrochemical plastic as feedstock.
Yes. Hi, everyone. My name is Eugenia, and I am one of the master students at the Charles Lab, or the only master student at the Charles Lab. And today I'll be talking to you about my project, which has to do with the biocatalytic production of PHAs using petrochemical plastics as a feedstock. And so there's quite a bit of overlap with the previous people that have gone before me, but I will be giving you, my project is also in the beginning stages, and so I'll be giving you a literature review as well as an overview of where the project will be going, or the direction of the research. So plastics are not a new material. We're all very familiar with them. It seems like they've been around for a long time, but they haven't. The first synthetic plastic was produced in 1907. And from that point until the 50s, there's a rapid increase in plastic production. And by the 50s, 2 million tons of plastics were produced. Currently, 400 million tons of plastics are produced, and that's a 200-fold increase in plastic production in less than a century. And majority of these plastics are single-use plastics that have a very short in-use lifespan. So they're used very briefly and then discarded, and this propagates a linear plastics economy. So it seems like plastic production is increasing, and this hints to the fact that it's become a very vital component of our global economy. And why is that so? Well, first of all, they have a low cost of production, but most importantly, they have excellent mechanical and thermal properties. And so they've surpassed the limits imposed by traditional materials like wood, metal, and glass. Unfortunately, the traits that make them so desirable also make them recalcitrant to degradation. And what this means is it will accumulate in the environment rather than decompose. Microplastics, which are plastics that are less than 5 millimeters in size, have garnered a lot of interest lately because their small size allows them to concentrate organic pollutants and then enter the food chain via trophic transfer, which can be which can impact the lives of living organisms that ingest these microplastics. So are there any waste management strategies for plastics? Yes, there are. We have mechanical and chemical recycling, landfilling, and incineration, but without going into too much details, these methods are not very sustainable. They pose threats to the environment and they are costly. And so researchers have turned to biocatalytic recycling, so using microorganisms as a technology to recycle plastic waste, because this is more environmentally friendly. So what does biodegradation of plastic look like? So the first step is a deterioration that's jointly accomplished by biotic and abiotic factors, and the goal of this is to expose more of the plastic's surface area for enzymatic attack. From here, we have biofragmentation, where the plastic polymers, the polymer chains are broken down into lower molecular weight oligomers and monomers that can then be assimilated and mineralized into products like carbon dioxide and water. So now I will talk about polyethylene terephthalate, which is one of the main plastics for my project. So polyethylene terephthalate, quite compared to the other types of plastic, I think research has seen more success in terms of using microorganisms to degrade PET, and majority of the enzymes that have been identified to play a role in polyethylene terephthalate degradation belong to the alpha-beta hydrolase enzyme family, and they consist of cutinases, lipases, and carboxyl esterases. But in 2016, a flagship example flagship example, I guess, of microorganisms that can degrade polyethylene terephthalate was isolated. And this organism is Ideonella sakaiensis 201F6. And this organism can use 1.9%, so low crystallinity PET film, as a major carbon and energy source. And it does so by producing two unique enzymes. So the first one is PETase, which converts PET into its intermediate MHET. From there, it produces a second enzyme, MHETase, that converts MHET into its environmentally benign monomers. Okay, so I think there's something wrong with my slide. There should have been two structures, so ethylene glycol and terephthalic acid. That's the breakdown product of polyethylene terephthalate. I'm not sure what happened there, but moving on. The next plastic that my project will focus on is polyethylene, and I think Jordan did a good job at providing a literature review as well as the enzymes involved, but I will go over that very briefly. So polyethylene terephthalate is considered more difficult to degrade than other types of plastics because it has a carbon-carbon backbone and lacks hydrolyzable functional groups. But I think regardless, research has seen more success, I guess you could say, in terms of using microorganisms to degrade polyethylene compared to other types of plastic. So perhaps they're a bit more degradable than we think. And in terms of its degradation, more than 20 genera of bacteria and fungi have been identified to possess this phenotype to be able to degrade polyethylene. And it has been suggested that the enzymes involved include ALK-B family of alkane hydroxylases, as well as lignin degrading enzymes like lacases, manganese peroxidases. So I'm going to pivot a bit and talk about Pseudomonas and PHA production, just to tie this literature review together. PHAs are essentially intracellular granules of carbon and energy storage. They're produced when essential nutrients are limiting, but carbon is in excess. They're categorized into two groups. So we have the short chain length PHAs that are between 3 to 5 monomers, and they're characteristically hard and brittle. And then we have the medium chain length PHAs, which are between 6 to 5 monomers in length, and they're more stretchy, so they're elastomers. Pseudomonas pitida KT2440 can produce medium chain length PHA, and this strain of bacteria is very popular in the industry. It's very robust. It can grow under some pretty harsh conditions, and there are many tools and technology used for its engineering. So it's a pretty good candidate for this project. And other researchers have engineered Pseudomonas pitida to be able to produce PHA using a variety of carbon sources like glycerol. So this leads me into my research objectives, which has to do with identifying novel genes and enzymes involved in polyethylene and polyethylene terephthalate degradation. And these genes and enzymes will be isolated from plastic-rich sites using functional metagenomics. And the goal of this is to use these genes to engineer Pseudomonas putida to produce PHA using these plastics as feedstock. So this will involve two steps. The first, I guess, part of the project will have to do with enriching for the relevant organisms, so organisms that can break down the plastic. But I think even before doing so, we're going to enrich for organisms that can break down the breakdown products. So in the case of polyethylene terephthalate, that would be terephthalic acid and ethylene glycol. Pseudomonas putida lacks the machinery for terephthalic acid metabolism. It can produce the aromatic acid like compound metabolite PCA or protocatechate, which feeds into the central carbon metabolism, but it cannot convert terephthalic acid into PTA. It doesn't have that machinery. And so we essentially be looking for organisms that can metabolize TPA, so we can use those genes to engineer Pseudomonas putida for this function. The second breakdown product of polyethylene terephthalate is ethylene glycol. And although Pseudomonas putida KT2440 possesses the machinery to break down ethylene glycol, two key genes are constitutively repressed. And so part of this project will have to do with enriching for microorganisms that can break down ethylene glycol or use ethylene glycol as a sole carbon source. And then the genes relevant for this function will be used to engineer Pseudomonas putida KT2440. Another approach that I've seen one paper take is to perform an adaptive laboratory evolution where they subject Pseudomonas putida to a very defined set of conditions. So growing Pseudomonas putida in media containing ethylene glycol as a sole carbon source. And what some of them have found was that, for instance, one paper saw that when they took this approach, the repressor for these genes, there was a mutation in the sequence, which resulted in a loss of function. And so this allowed Pseudomonas putida to be able to use ethylene glycol as a sole carbon source. And so after all of this, like Jardin said, working backwards, we're then going to enrich for microorganisms that can degrade the plastics that I have mentioned. Actually, they'll be done in parallel, but the engineering portion will focus on the breakdown products first before using the genes that we identify for plastic degradation for subsequent engineering. So after that, it will follow the regular functional metagenomics workflow where we extract the DNA from the enrichment cultures. We construct metagenomic libraries. We express these libraries in Pseudomonas putida because, as I mentioned, Pseudomonas putida cannot metabolize or break down any of our substrates of interest, so our breakdown products and our plastics. And we are going to screen for complementation. And so we're going to be looking for clones that confer terephthalic acid utilization or ethylene glycol utilization, or in the case of polyethylene, a short length alkane like hexadecane or the plastics. We're going to screen for complementation by growth. So growth on these substrates will be indicative of a clone that possesses a gene relevant for this function. And from there, they'll be sequenced, assembled, and annotated to predict the gene functions. So the bigger picture of this project is to take microplastics, use that as feedstock for PHA production, taking the PHA to produce plastic products that are inherently biodegradable. And when they're used and discarded, they can be broken down as like a source of carbon and energy for the production of more PHA, which can then be used to make more plastic products. And so that feeds into a circular pathway, which will hopefully eliminate the linear plastics economy that we see currently. So yeah, thank you so much for listening to my presentation. Thank you to Dr. Charles, my supervisor, for his guidance on putting this together, and for my lab mates as well for their input along the way, and for the NFRF for the funding for this project.
Any questions? I'll ask one. Do you think it will be more of a challenge to find the enzymes to break down the plastics, or to engineer the bacteria to use that as a carbon source and then produce new biopolymers?
Yes, that's a good question. I think the difficulty levels might be the same, just because I have encountered a paper that was recently published where they tried to do something similar. So that first strain, Legionella sakiensis, that can break down PET into its intermediate and then its starting monomers. They try to engineer it to break down plastics using enzymes that have already been discovered, and also to engineer it to break down products. And what they found was that they weren't able to achieve what they call PET trophy in both, like in Pseudomonas putida, like as a whole, because there were a bunch of factors coming into play, where one of them was that you need, like, the balance between enzyme expression and nutrient availability. You need a certain level of terephthalic acid and ethylene glycol to be able to produce the enzymes, and you need a certain level of enzyme expression to be able to break down polyethylene terephthalate to produce the terephthalic acid and ethylene glycol. So I think that part, just based off that paper, shows that that might be one hurdle. But I think, yeah, just in general, we're hoping to see or to find genes that can break down plastics, because the sources that we're looking at, one of them is a landfill. So the plastics there have been in contact with the soil for an extended period of time. So I can't really tell, but hopefully just based off of yeah, these factors, I'd say similar level of difficulty, in my opinion.
Thanks. Any other questions?
Hey, maybe just a general question. Do you think that it will solve a lot of problem if we increase the recyclability of plastic in Canada into 60% or something?
Sorry, could you repeat that?
Because for now in Canada, you mentioned that it's only 9% of plastic is being recycled.
Yeah.
But if you can able to
Increase it to 60%.
for some.
I think to some extent, but due to the nature of plastics, I think recycling can only reach a certain extent. You can't, for some of the plastics, you can only, for instance, like the mechanical recycling aspect, one of the downsides is that the products that are produced are of lower quality than, like, if you make it with the virgin PET or like the virgin plastic themselves. So I think we're going to need to complement that recycling with other forms of plastic waste management. That might not be, might not solve all of the problems, because I think even recycling, mechanical and chemical recycling itself, they have their own problems, which is why researchers are looking to other alternatives, if that makes sense. Did that answer your question?
Yeah, sure.
Okay.
And so do you think that is the problem is the of microplastic is more important than enzymatic degradation of, for example, the PET bottle?
The PET bottles? I think they're both equally important, but with like the nature of my project, the focus is microplastics. But I think once we're able to, I guess, crack the code or find something that works, maybe that can be extended to larger pieces of plastics, where maybe they can be crushed into smaller particles, because surface area is, I think, one of the factors that affects degradation. And so maybe we can extend it to plastic bottles, although, like you mentioned earlier, most commercial plastics have additives in it that might interfere with the degradation process. But I think generally, maybe it can be extended to larger whole, like, plastics.
Thank you.
Thanks. So we'll stop there. Thanks again for the talk. We'll have Aranksha Thakur talk about functional metagenomics enabled engineering of Pseudomonas for the production of PHA copolymers using lactose.
So my name is Aranksha. I am also a part of the Charles Lab, and my presentation focuses on polyhydroxyalkanoate or PHA production, PHA copolymers using lactose as a feedstock. So bioplastics overall is just an umbrella term that refers to plastics that are biodegradable and or made from renewable resources. Polyhydroxyalkanoates are a type of bioplastic in that they are biodegradable and derived from renewable resources. They're produced as a reserve carbon source by the bacterial cells. So the bacteria produces this polymer under nutrient limiting conditions in granules that are referred to as carbonosomes. PHAs are very diverse in their thermal and mechanical properties, which is why they're appealing as an alternative to fossil fuel-based plastics. This diversity comes from a diverse range of monomers that can be potentially polymerized in this polymer, so there could be a mixture. The most common monomers are referred to as short chain length or medium chain length. And this is just depending on the chain length of the variable group. The short chain length monomers are 4 to 5 carbons, and the medium chain length are 6 to 12. The enzyme that synthesizes this polymer largely determines what monomers are going to be in the final monomer. So the current challenges or barriers to PHA being more widely adopted is the production cost and also the incompatibility with some of the uses that commercial plastics currently have. Most of the cost problem is dependent on what feedstock is used. So recently people have shifted the focus on using lower cost feedstock, such as food waste, to produce PHA. The food waste stream that we're focusing on is lactose, which is produced during the production of lactose-free milk and is very common dairy waste. So in this case, food waste can also be a negative cost feedstock because it's accompanied with waste management problems. Another conflict is that PHA homopolymers, so exclusively short chain length or medium chain length polymers, have conflicting properties in that short chain length polymers are hard and brittle. So they have a really good mechanical strength, but they aren't very tough. They're really susceptible to fracturing, while MCL PHA are soft and ductile. They're almost rubbery, so they can't maintain their shape well. But combining short chain length and medium chain length PHA will more likely make the polymers closer to having the mechanical and thermal properties that are similar to traditional fossil fuel-based plastics, such as polyethylene and polypropylene. And some of the applications that PHAs have already been used in do rely on copolymers. So the core objectives of this part of the project were to first engineer a strain with all the genes that are already predicted to be involved in utilizing lactose and galactose, and integrate these into the genome and to test them. This was followed by, so this involved just starting with LAG, Z, and Y for lactose utilization, very well-characterized genes, and then followed by the integration of the delayed Dodorov pathway for galactose utilization, which has already been shown to allow galactose utilization in Pseudomonas alopatita. So following this, I performed a functional metagenomic screen to isolate genes that improved galactose utilization. And then lastly, different PHA synthases will be introduced into the lactose utilizing strains to see what copolymer compositions we can achieve using the lactose feedstock. So integrate these genes into Pseudomonas aliputida KT2440. I used a counter selection based method using the gene SAFB to integrate these genes into our choice of strain. The final strain this resulted in was PPUW44. So growth experiments on this strain show that lactose is being used well, comparable to glucose. So lactose is in gray and glucose is in green. But it does not use galactose as well. So we decided to look for other genes that might improve galactose utilization in our strains. So to do this, we use a simple metagenomic screen by transferring a metagenomic library. The one that we're using is called 11AW. It was constructed by using isolated DNA from wheat field soil. After the transfer, we plated the mixture on minimal media containing galactose as a sole carbon source. So selecting 4 cells that have a cosmic clone that then allow KT2440 to grow on galactose. We then transferred these clones back into E. coli to be able to isolate the cosmic DNA. This method generally isolates many duplicates, so we have to determine which clones are unique first. And then once we identified the right clones, we sent them off to be sequenced. So doing this, using this library, I was only able to isolate 1 clone. Based on its gene function annotation, there are genes that could be involved, or they're predicted to be involved. 3 of these, DGO-A, DGO-K, and DGO-D, the first 3, we've already included in our original strategy. There is also a galactinoid transporter and a galactinoid regulator. I also determined the possible origin of this DNA to be Pseudomonas fluorescens using Philo-P. So just to test the function of this clone in our already engineered PPU like KT2440 strain, I conjugated it into the strain, and it looks like growth on galactose is significantly improved in the strain with this clone, but growth on lactose remains the same. Not sure why this is happening. It might be because I'm using excess lactose in these experiments, but it does seem like some of the genes involved, it does seem like growth is improved on galactose, so the next step would be to figure out which ones are involved. So just to recap a bit, the enzyme responsible for polymerization determines the monomer composition of the PHA that the cell will produce, because it has a specific substrate specificity for a certain type of monomer. The diversity in monomer composition can be achieved by using metagenomically isolated PHA synthases. So a member of the Charles lab, Dr. Chang, has previously isolated a wide range of PHA synthases. He was able to showcase that once he introduced these cosmic clones into the same host, KT2440, he was able to showcase a wide range of monomer compositions, both short chain length, medium chain length, and then copolymers as well. But this was done using fatty acid carbon sources, so I'll be doing the same using lactose as a carbon source to see what we can achieve. So to start off, we just introduce the clones in. Eventually, we'll want to put these into the genome, but to start, I removed the native PHAC operon and replaced it with an RFP gene, just so the native PHA production of native PHA production of Pseudomonas putida doesn't affect results. And then put in 3 cosmic clones to start, and research at Lambton College, Alexa Galbraith and Riddhi Patel were able to figure out the monomer composition for us. They do produce both, the first clone 14, produces only short chain length monomers, but clone 16 and clone 20 produce both short chain length, medium chain length copolymers. So the next step would be to determine what the mechanical properties of these copolymers are, and what we could potentially use them for. So the future steps would be to determine just which genes on the clone are responsible for galactose utilization. For this, we'd use transposon mutagenesis, either in vitro or in vivo. And then also start integrating these PHA synthases into our lactose utilizing strain for stable expression. So then we could use these at a larger scale. And thank you for listening. That's the end of my presentation.
Any questions for Amsha? Okay.
Thank you for a nice presentation. I got excited. I saw some carbon metabolism. So just, I might have missed it in the presentation. So are galactose and lactose both incorporated in central carbon metabolism through the delayed Dudroff pathway in this strain, or?
Yes, so lactose would be broken down into glucose and galactose, and then the galactose would be utilized or by the delayed Dudroff pathway.
Okay, so it enters at the same point.
Yes, so the glucose will enter its own pathway, and then the galactose will enter the delayed Dudroff pathway.
Okay, thank you.
Once you've introduced the PHA genes into your model organism, you're going to have a major shift in the carbon flow in the system. Do you expect that to have any secondary effects on the physiology of the organism? No, unexpected effects.
Sorry, the physiology?
Well, yes. So, for instance, as has been seen as in, I think, lots of other examples, when you shift carbon flow, the bioethanol studies, you suddenly start finding problems with deficits in nitrogen metabolism, for example, and all sorts of other things. I just wondered if you'd contemplated that or read much about it, and whether you might expect to have some similar effects when you do this.
Yeah, I'm not sure. That's a good question. Sorry, you could continue.
I mean, it's the unexpected things.
Right.
So you haven't done it yet, so you don't know what's going to come up. It might be worth reading the background of those groups and industries that engineered, for example, particularly organisms for bioethanol production, where they were trying to shunt all the carbon in the system,
right,
substrate through to a single product, and then discovered that all sorts of other metabolic effects they weren't anticipating suddenly started happening.
Right.
So, deficits in amino acids, if I remember one of the stories I read, carbon that would normally go into a key amino acid biosynthesis is now going somewhere else, being directed down to a product. And so you are having negative effects, physiological effects on the organs.
Right.
Great. I don't see any more questions. We'll stop there. Thanks again for the great talk, and thanks to all of our speakers this session. Yeah, so that's the end of the session. I don't know if Trevor, if you wanted to say stuff or for the poster.
A great end to the first day and the final day for the virtual session. So I know that there are still some attendees who are present virtually. I thank you for your participation. This is our first attempt at a hybrid event, and I think it went pretty well under the circumstances. So thank you for your participation. We hope to see you in person at the next in-person functional metagenomics meeting, and have a good rest of your day wherever you are in the world. Thank you very much.